Hi, my name is Richie and I will be introducing DnaB, a bacterial helicase. DNA replication involves an intricate coordination of a large collection of proteins that each fulfill specific functions. A critical step is the unwinding of helical DNA so that complementary strands can be built. DNA helicase is responsible for this unwinding process. Here we focus on the helicase called DnaB, which is important in bacterial DNA replication. DnaB is built from 6 identical monomer subunits arranged in a hexagonal shape. Each subunit has a C-terminal domain and an N-terminal domain, which are connected by a linker domain. The CTD contains binding sites for NTP molecules that supply energy. The 6 subunits then form a CTD ring, which contains 6 DNA binding loops. These loops form hydrogen bond networks that anchor DnaB to single-stranded DNA. The NTD is a helical bundle that terminates in a helical hairpin and has also been proposed to bind ssDNA. Each DnaB subunit either places its helical hairpin on top of its own CTD or the CTD of an adjacent subunit. This gives rise to a characteristic trimer of dimer structure in the NTD collar. Overall, DnaB forms a ring-like spiral staircase around single-stranded DNA. In this form, the CTD ring can be viewed as a 6-step staircase, while the NTD collar forms a 3-step staircase since it's a trimer of dimers. DnaB helicase moves along a DNA strand in a hand-over-hand fashion, just like climbing a rope. Double-stranded DNA enters DnaB from the CTD ring, and a pin or wedge occludes the 3' leading strand. On the other hand, the 5' lagging strand can freely pass and exits through the NTD collar. Fundamentally, this translocation mechanism involves the sequential movement of individual subunits along the helical axis. For each hydrolysis event of one NTP molecule, the CTD makes a step size of 2 nucleotides towards the 3' end of ssDNA. Movement of the NTD collar is closely coupled to CTD movement. Particularly, each dimer of the NTD collar moves into a new position for every 2 CTD movement events. It's thought that each dimer remains intact while moving due to a large internal hydrophobic surface.